Just want to start by thanking Children's Cancer CoLab for their generous support of our work. The funding is really enabling us to accelerate our discoveries to help develop new approaches, specifically to try to strengthen the immune system post-stem cell transplant in pediatric cancer patients to hopefully prevent infections. I'm excited to have this opportunity today to talk a little bit of an overview of our work and discuss some of our research goals. So we're very interested in trying to understand how our cells make blood, and this is highly relevant in the treatment of cancer patients in both adults and children. So in our bodies, stem cells in our bones make all the blood cells. And this includes red blood cells that carry oxygen around the body, platelets that are required for clotting, and also all the immune cells that we use to fight infections. And our bodies make billions of blood cells a day, and this is required for us to function. So in cancer treatment, cancer therapies can often severely damage our blood system. And also some cancers specifically also come from abnormal blood cells. So the treatment involves targeting normal and abnormal blood cells. And an example of this might be leukemia. So we're really interested in trying to understand hematopoietic stem cell transplants and how they're used as therapy to treat cancer patients. And this is a type of cellular therapy that we investigate in the laboratory. So stem cell transplants involve high-dose conditioning so that the blood system is damaged, and this allows the cells, the cancer cells, if they're in the blood system, to be treated, but it also allows the engraftment of donor stem cells. Now, there's a period of time where these donor stem cells then give rise to blood lineages in the body. And so different blood lineages actually develop at different rates. So innate blood cells such as monocytes and neutrophils generally recover quite quickly. However, our adaptive immune cells, such as our T cells, can take 6 months to years to recover properly. And in particular, T cells are one of the last blood lineages to recover post-transplant. In infants, this can be between 6 to 9 months. In children, this is 12 to 18 months, and in adults, this can be years. So after hematopoietic stem cell transplants, one of the major risks of morbidity and mortality is severe opportunistic infections due to a compromised immune system. And this is particularly because of the impaired T cell immunity. And viruses that become particularly life-threatening in this context are things like CMV, EBV, and adenovirus. So not only is the body unable, because it's immunosuppressed and is not making T cells, is unable to control infection, but also the treatments used, antiviral treatments, also have their own immunosuppression and toxicities associated with them, making this a major risk of morbidity post-transplant, particularly early. And there's a reason that T cells are one of the last blood lineages to recover. So as I mentioned before, stem cells sit in our bone marrow, and these stem cells make all the blood lineages. And these blood lineages are made in a factory. It's just the sort of spongy part of inside our bones. And all the lineages, except for T cells, are actually made in the bone marrow. And what we know about T cells is a very rare subset of these stem cells has to form these very specific thymic-seeding progenitors. They then leave the bone marrow. They migrate through the bloodstream where they enter the thymus, which is the organ where T cells develop. And this is a long process. And it's also a population of cells that we really don't know much about. because they're very infrequent, they're very hard to study, and particularly them being a rare cell population. And so that's one of the key research questions that we have to try to understand how thymic-seeding progenitors develop. So we might be able to speed up the development of T cells and find new ways of making T cells from transplanted cells. And so we have a number of research questions. So first of all, can we find and characterize the best cells to transplant to rapidly make T cells? Can we identify molecular targets that might help us to improve the ability of cells to make T cells? And also, can we develop these targets as therapies to enhance T cell recovery in transplants?
But all cells in the body have the same DNA. What makes them different is the genes that are expressed or turned on in each cell. In the blood system, all mature blood cells, such as red blood cells and immune cells, originate from blood stem cells. And although these stem cells may look identical, they're actually not, and individual cells express different sets of genes. These subtle differences can influence how each cell behaves and what type of cell it will ultimately become. So the key question we're trying to answer here is, how do stem cells decide on their fate and which genes regulate these decisions? So to address this, we need technologies that allow us to both examine the gene expression in individual cells and also track their behavior over time. So one powerful tool we use is called single-cell RNA sequencing. Genes that are switched on produce RNA. By measuring RNA of individual cells, we can determine which genes are active in each cell at a given time. This is a major advance over previous methods, which measured gene expression across large populations of cells simultaneously, averaging their signals. A useful analogy is comparing a fruit smoothie to a fruit salad. Bulk RNA sequencing is like a fruit smoothie where everything is blended together and individual fruits are indistinguishable. In contrast, single-cell RNA sequencing is like a fruit salad where each piece of fruit remains intact, allowing us to examine them individually. This is really important because even cells that look identical can actually behave very differently. While single-cell RNA sequencing is very powerful and can tell us what a cell is doing at that moment, it is equivalent to taking a single snapshot in time and does not show us where that cell came from or what it will become. So to overcome this limitation, we use a complementary method called lineage tracing. So in our lab, we've developed a method that introduces a large library containing millions of unique DNA barcodes into cells. Each cell gets a distinct barcode, which is stably inherited as the cell divides. As a result, all progeny of a given cell carry the same barcode. By analyzing these barcodes, we can reconstruct the lineage relationship between cells and map which stem cells give rise to which type of blood cells. By integrating lineage tracing with single-cell RNA sequencing, we can link a cell's fate with its gene expression profile. This allows us to determine not only which cells are active, but also how these changes in gene expression control cell fate decisions. Combining these methods, we have looked at the genes that are expressed in thousands of blood stem cells grown in a dish. We show this data in a plot called a UMAP, which is a two-dimensional representation of single-cell of the gene expression of individual cells. So in this UMAP, each dot here represents a single cell. The cells which are more similar are closer together on a UMAP, and those that are more different are further apart. The most immature stem cells in red are at the top of the UMAP, and these develop into more mature types of blood cells in the lower part of the UMAP. We can look at the genes that are expressed in cells in each part of UMAP to work out what type of blood cells they are. We can also use a method called RNA velocity to predict how cells are changing based on how their gene expression is changing. Using our lineage tracing barcodes, we can see what distinguishes the stem cells that transplant well from those that don't transplant well. When we compare the gene expression, we found that stem cells that transplant well were much better at staying a stem cell after they divide and maintain their stem cell identity. To answer our question on what makes stem cells become T cells, we looked at the barcodes of cells that turn into T cells, then look at their gene expression by single-cell RNA sequencing. By comparing the gene expression of stem cells that make lots of T cells with those that don't engraft well and make many blood cells, we can identify the genes that control blood cell development into T cells. After we found the genes that may be important for T cell development, we can use them to identify the best stem cells to transplant that will engraft well and make lots of T cells. We can further study these genes to work out exactly how they function to control engraftment and make T cells. Ultimately, we want to be able to use these genes to enhance T cell recovery after transplant, so we've been optimizing methods to express genes in blood stem cells. One approach we've been using is lipid nanoparticles, which consists of RNA encapsulated in a layer of lipid molecules. These plots here show the result of an experiment where we induced the RNA for fluorescent protein. The level of fluorescence in cells is shown on the x-axis, and the y-axis shows the level of a dye that sticks to the cell membrane. The control cells without the lipid nanoparticles on the left, and the cells treated with the nanoparticles on the right. We can see that we can successfully deliver RNA to blood stem cells to express a fluorescent protein. After we found the genes that enhance T cell recovery, we will use our gene delivery system to express these genes in blood stem cells. Ultimately, we hope that our work will lead to a more rapid recovery of the immune system following hematopoietic stem cell transplants and improve the outcomes for children undergoing cancer treatment. We'd like to thank the many people that have contributed to this project, and of course the generous funding from the Children's Cancer CoLab that have allowed us to do this work. We're really looking forward to working with the Patient and Family Advisory Committee to continue our research.